of China. And it's there's been some recent expansions of new occurrences in parts of Europe and in the US. Okay, so our group was interested in what is the predicted distribution of this parasite and what are the factors that influence that geographic range. Where might we find it? Where is it going? So we can look now before people have diseases in a decade. So this is a quick talk, but we compiled a lot of parasite data, got relevant covariates, did spatial modeling of a species distribution model in R, and then projected that into a map, which we'll show. So this group is probably very attuned to all the different biodiversity data out there. We were limited because of our task. We needed to link it to environmental variables. So we could only use occurrences contemporary 1990 till like 2020, a 30-year period. This eliminates a lot of natural history collection data. Also, we needed fine scale location data. So there are some great host parasite data sets out there, but they might only be linked to a country or an area that's larger than 10 kilometers. So we really limited to what we could verify in the primary literature, and that met our specific use case. So assessing hundreds of papers, we found 363 presence locations, and we split these into testing and training data sets. And you'll notice there's some spatial biases. These are especially in Russia, where we could not get any primary literature. And there's an overabundance in Europe, where there's some fantastic parasite societies that have totally open access journals, like Hungary, France, Austria, and the Slovak Academy of Sciences, like a really rich corpus of health data for animals and humans. And unique to our data set, we have pseudo-absences from other helminth studies that did not detect E-multi. So these are wildlife studies where they were doing all those similar diagnostic procedures, but they did not discover E-multi. And this is much more widespread. It goes down in southern latitude, but you see a lot of the same spatial biases in those disease surveillance studies as well. So jumping through, what was months of work, we used several different open access data sets to compile climate, land cover, and host data. Interestingly, from our literature search, we established host species and then used those host species as our biodiversity component, filtering IUCN for those 53 wildlife species. Some factors that we probably would like to use but didn't find at the scale we needed were things like domestic dog distribution, fur bearer abundance, or fox abundances. These are kind of tricky areas of these domestic animals that might not traditionally be in what we think of as wild biodiversity. So we have built a really robust model where we were able to explain 97% of our testing data set, and climate, the rodent richness, and grassland were especially important in predicting this. From that model, we were able to then reproject over the Northern Hemisphere areas that have a propensity to have this parasite. And we saw some of those common endemic areas like the Tibetan Plateau and Northern Alaska, but also areas where there's a potential for great expansion of the wild hosted parasite. Okay, clicked, but okay. So we also identified some new susceptible areas that would be prime for new wildlife surveillance, particularly in the US and the Midwest, but also in the Caucasus. Armenia just had their first detection of E-multi in a canid this year. And so especially areas of like that, as well as Kazakhstan and Mongolia that we didn't have any primary data from, are areas to look for. 2 minutes, okay. So to finish with some kind of wrap-up thoughts, this was it's a complex life cycle parasite. And so this really takes a transdisciplinary team. We were a group of ecology students. We really benefited from talking to veterinarians about the diagnostics. Even though like this is a gastrointestinal parasites, I'm more familiar with huge tapeworms that are very large. This is actually a really tiny parasite that's hard to detect. And so incorporating that in filtering for our pseudo-absence data was really important, as well as working with our local librarians. Getting access to the primary literature is not a negligible task. That takes hours of work and really dedicated work. And we benefited from being at a university where they could do interlibrary loans, where we could request disparate literature. But even with that, a lot of the articles we wanted, we could not get. And so we know that there's been much more surveillance done, particularly in Russia, that we just cannot get access to that primary literature. We had a very specific use case. We wanted to do a prediction. So much data ends up on the cutting room floor from spatial thinning, for matching covariates, but we have tried to maintain our database so other people can use it and investigate future things like host competence and other aspects that might be important in new research directions. And yeah, to that end, the literature that we found, we are hosting on Zenodo. Our data is hosted on Figshare, but this is not our field data. So we're kind of in this weird space of where do we publish it? How does it get into GBIF? Our archives don't go into GBIF. So it's kind of tricky to put these literature-derived places into the biodiversity platforms that we're so familiar with. So with that, I'll thank the Center for Ecology of Infectious Diseases, the working group, and libraries. We also ended up even working with the university library, getting a ton of literature through Sci-Hub, which, I mean, if that's the last resort, that's the last resort. And with that, I'll take questions.
I have many questions, but they're all good things. So thank you for a very good presentation. I have a background in niche modeling, so from what I can tell, seems to be very thorough. But more specifically, I am a science officer at GBIF, and would love to talk with records if you're interested in sharing, think about how to share data.
Okay, great. Yeah. So we have an officer from GBIF who offered to maybe help with the nuances of literature-derived data into GBIF. So yeah, excited about that.
Yeah, it was similar because there are cases of data sets, data taxon occurrences taken from references from thousands of papers, including Russian papers, and published twice a year, say.
You just did. So yeah, no problem.
So Francis Banwell, who's the next talk, who will introduce himself.
Yeah. So hi everybody, I'm Francis. I'm a postdoc at Université de Montréal in Quebec, Canada, and I'm also a member of the GeoBond One Health Working Group. So does anyone know what is the GeoBond One Health Working Group? No? Not even like the members? Okay. So basically, the GeoBond One Health Working Group is a working group that aims to mainstream the health of ecosystems and the environment into the One Health approach. And one of the things that we do is that we try to assess what we can use from biodiversity domain into the health domain. So the motivation behind our work is the ongoing recognition that the health of humans is linked to the health of animals and the environment. So for example, there was the CBD, who adopted the global plan on biodiversity and health, which has a bunch of actions that's trying to mainstream the interlinkages between biodiversity and health. And more recently, there was the WHO Biodynamic Agreement that also recognizes the interlinkages between biodiversity and health, but this time related to zoonotic diseases. Moreover, there's the One Health Joint Plan of Action that was adopted by the quadripartite organizations in 2022 that recognizes the link between the health of humans, animals, plants, and the environment. So this plan has a bunch of actions in different action tracks to try to tackle different problematics on zoonotic diseases, but also food safety risk and antimicrobial resistance. However, there are currently no indicators for monitoring these actions in this action plan. So our work is to try to see if we can reuse existing biodiversity indicators for monitoring these actions. So what we did is that we went over the indicators in the GBIF. So there's 204 indicators that we assessed. And we assessed first if they were linked to any aspects of health. So more specifically, for each indicator of the GBIF, we assessed if they were either directly linked, indirectly linked, potentially linked, or not linked to human health, animal health, plant health, and environmental health. So I'm going to give you two examples of indicators that are directly linked to every aspect of health. And the first one is headline indicator 7.2, which is the pesticide environment concentration and our aggregated total applied toxicity. And by reading the definition, we see that this indicator measures the risk to ecological communities related to pollution. So this indicator is directly linked to every aspect of health, so to human health, animal health, plant health, and environmental health. A second indicator is component indicator 11.CT.2, which is the proportion of bodies of water with good ambient water quality. And what we mean by good water quality is ambient water quality that does not damage ecosystem function or human health. So this indicator is also linked to every aspect of health. However, it's not every indicators that are linked to every aspect of health, but overall, we found that 75% of GBIF indicators were either directly or indirectly linked to either animal, plant, human, or environmental health, which is a lot. So this justifies the second part of my presentation, which is about to see which one of these indicators could be used to monitor specific actions in the One Health Joint Plan of Action. So this is what we did here. So for each indicator, we assessed if they were either directly usable after adaptation, or not usable for monitoring specific actions in the action plan. But before going into the details, just going to go over what is the joint plan of action. So there are 6 action tracks on different problematics related to problems at the intersections of human, animal, plant, and environmental health. For example, the first one is about health systems and capacity building. The second one about epidemics and pandemics. The third one about neglected tropical and vector-borne diseases. The fourth one about food safety risks. The fifth one about antimicrobial resistance. And the last one about environmental health. So what we found, so before for each indicator, we assessed if they were usable or not for different actions in the action tracks. So here are our results. So here we see that a lot of indicators could be used to monitor each of these action tracks. We find, for example, that 80% of indicators could be used to monitor the action track 6 about environmental health, which makes sense because biodiversity indicators, they measure something related to environmental health. However, action track 5 about antimicrobial resistance, only 20% of indicators could be used to monitor this action plan, these actions. So we see that even though a lot of indicators could be used, we see that there are still some gaps. Next, we disaggregated this analysis into different types of indicators. So here we see for headline indicators, we found that 100% of them could be used to monitor at least one action in the action plan. For the binary indicators, it's 93%. For the component indicators, 94%. And complementary indicators, 87%, which is a lot. So here also what we see is that if we count the numbers, we see that most indicators that could be used are either component or complementary indicators, which are optional indicators. So that means that parties don't necessarily report on them. So to answer my question, can we reuse existing biodiversity indicators for monitoring One Health actions? The answer is yes, but GBIF indicators don't equally cover all action tracks. And also a lot of indicators are optional, so this doesn't mean that they will actually be measured by the parties. So I just want to finish on this note that One Health is a collaborative effort. And if you want to make sure that we reuse our knowledge of biodiversity, we can take indicators from the GBIF, but also indicators from other agreements. Thank you. Do you have any questions? Can also be in Spanish, and my Spanish is not really good. Actually, it's very bad, but I try to. Do I have time for a question? Yeah?
Yes,
Yeah.
I'm not Mike, so.
Oh yeah, I need to. No? Okay, sorry, I still need to.
What would be like the best indicator? I mean, I'm curious about the indicators and what do you think, what's going to happen? What's the next step? What do you propose? What are people going to do in order to follow up on your work?
Okay, so this was just an exploratory approach. So we don't have any mandate from the ecology organizations to do this. So our work is just to try to, identify and tell people that this is a possibility to do, instead of like reinventing the wheel, we can find indicators that already exist to monitor actions in the action plan. Sometimes it needs some adaptation, but it's just like a way to minimize the work for parties. Yeah. Any other question? No? Yeah.
Discuss more later.
I get you.
Okay. No, actually, it's we have a virtual talk now from Solidad. Excuse me for my Zakaria. Oh, you're here. Okay, welcome. So it's virtual. Yeah. Ben.
Here.
Introduce yourself.
Okay, thank you. Well, I'm going to speak in Spanish. So if you need a translation service, you have this crowd. Bueno, buenos días. Mi nombre es Soledad Secarelli. Trabajo en el laboratorio de Tratominos en Argentina. Y voy a presentarles un trabajo en el que articulamos cuestiones sobre cómo hacer que nuestros datos sobre biodiversidad de tratominos, que son los vectores de Chagas, pueden llegar a ser importantes para articular redes científicas, colaboraciones interinstitucionales y la ciencia participativa. Bueno.
Yeah, the one down at the bottom.
No,
Justo conmigo. Practice Spanish. Ah, me habías prometido que funcionaba. Gracias.
Bien.
Ok. Bueno, entonces, para empezar, una introducción que es Chagas. Nosotros como grupo también tomamos al Chagas no solo como una enfermedad, sino como una problemática que están involucradas un montón de dimensiones y por eso lo tomamos como una problemática compleja multidimensional en el cual se entremezclan diversas dimensiones biomédicas, epidemiológicas, socioculturales y políticas. Pero sabemos que a nivel epidemiológico hay alrededor de entre 6 y 7 millones de personas infectadas a nivel mundial. Si pensamos en las vías de transmisión del parásito que ocasiona el Chagas, que es el Trypanosoma cruzi, hay diferentes vías de transmisión. Está la vertical, hay algunas por transfusiones sanguíneas o algunos órganos que pueden estar infectados, la ingesta de algunos alimentos o bebidas contaminadas, accidente de laboratorio, pero una de las principales son nuestros triatominos, que por medio, cuando se alimenta y defeca, en su materia fecal, está el parásito y puede ocasionar o no la enfermedad de Chagas. No todas las personas que se infectan desarrollan la enfermedad. Entonces, bien, hablando de los triatominos, nosotros nos encargamos de recopilar información de las 159 especies que están distribuidas en todo el mundo. Las principales y la mayoría están en América. Tenemos 143 especies distribuidas desde el sur de Chile y Argentina hasta la mitad de Estados Unidos. Y el resto de especies están en los continentes de Asia, de Oceanía y de África. Acá quedó cortado y dice solo Asia, pero son los 3 continentes, que son uno de los géneros se comparte con América, que es el triatoma, y el resto es otra especie que está solo allá. La diferencia es que en Asia, Oceanía y África todavía no está el parásito, pero están los vectores, y hace muchos años que están los vectores. Y después, para que vean, los países que están marcados con verde son países que tienen personas infectadas con cruzi. Y como veíamos recién, el ser humano es una fuente de alimento de los triatominos y a su vez una fuente de circulación del parásito. Entonces, recopilamos esa información de una manera de tratar de agarrar un montón de fuentes de información, desde repositorios públicos, como recién mencionaban, nuestros propios trabajos de campo y los trabajos de campo de nuestros colegas de todo el mundo, porque tenemos colegas de América, pero también de China, que nos han aportado los datos, y para juntarlos. Y también la ciencia participativa o la ciencia ciudadana, que es un proyecto que tenemos también desde Argentina, se llama GeoBin, por bin chuca le decimos nosotros a los triatominos. Y bueno, están separados en esos 3 conjuntos de datos por una cuestión de logística. Un set de datos tiene a las especies que tocan suelo argentino, otro set de datos tiene el complemento de especies americanas, y el tercero tiene todo lo que es fuera de América, que es el último más nuevo que tenemos. Bien, entonces, ¿cómo hacemos para recopilar los datos a través de la ciencia ciudadana? Desarrollamos una aplicación de la cual, si alguien encuentra un insecto que piensa que puede llegar a ser un triatomino, le puede sacar una foto de arriba, una foto de abajo, por las características que nos ayudan a diferenciar las especies, y la envía. Selecciona en qué hábitat la encontró por una cuestión epidemiológica, y se geoposiciona con el celular mismo. Eso nos llega a este panel de control que tenemos acá, en el cual podemos en Argentina clasificar a qué provincia pertenece ese dato, y ahora lo estamos ampliando a otros países. Estamos ahora trabajando justo con una colega de Brasil, y tenemos algunos otros países que vamos a empezar a hacer pruebas piloto, pero la cuestión no queda solo en que nosotros respondamos si es vinchuca, triatomino, no es triatomino, sino que si es un triatomino, conectamos después con los ministerios de salud de cada provincia. En Argentina ya tenemos, de todas las provincias que tenemos, a 20 de los agentes sanitarios que están en cada provincia capacitados para poder responder a través del panel de control de GeoBin, y que ese dato quede registrado, pero que se dé una respuesta sanitaria. Porque por lo menos en Argentina, por ley, hay que dar una respuesta sanitaria. Y no solo queda ahí, sino que la articulación que hacemos con los ministerios de salud a nivel nacional, pudimos realizar este catálogo, que lo tengo por ahí si lo quieren ver, que es práctico, es de bolsillo, y es para que los técnicos de campo puedan diferenciar fácilmente a los triatominos que se encuentran y también poder ayudar en esa respuesta que se da a la comunidad. Así que bueno, esta es una colaboración interinstitucional importante que tuvimos. Y bueno, si seguimos juntando, esta es la red de triatominos que estamos coordinando con un colega del laboratorio, que hasta el momento vamos 12 países, porque se sumó un país más, que es Perú, así que América relativamente está bastante representado, estamos trabajando con más de 24 instituciones, y lo que hicimos es llevar todos estos datos a desarrollar esta plataforma que está saliendo ahora, en el cual cada uno de los que trabajamos con triatominos podemos acceder a mirar especie por especie su distribución, y si necesitamos ese conjunto de datos, conectamos con los set de datos de GBIF que recién les comentábamos, y estamos viendo cómo articular con eNaturalist, porque hay muchos colegas brasileños que están en eNaturalist respondiendo, pero creemos que necesitamos juntarnos y trabajar mejor en articular las respuestas hacia ahí, porque es un afluente muy grande de información, pero como trabajamos con insectos de interés sanitaria, queremos tener cuidado, pero bueno, las conexiones están, los contactos están, y bueno, esta plataforma ya casi está lista. Ya terminé. Así que bueno, esto es como el filtrado de esa especie que es Triatoma infestans, la especie que está en el cono sur, que es la más importante para nosotros, y todos esos datos es lo que se ve ahí, como las personas van, filtran y se quedan con ese conjunto de datos de esa especie, en esa plataforma. Así que bueno, muchas gracias, todo esto se hace en equipo, y bueno, acá están las colaboraciones, el nodo argentino, bueno, y el financiamiento que tuvimos, que sin ello no podríamos hacer nada. Muchas gracias. Thank you. Yes.
Gracias, Soledad. Me gustaría, pues, bueno, primero que todo me parece un trabajo súper interesante desde la vigilancia entomológica. Me gustaría saber de pronto qué desafíos han encontrado para llevar a cabo esta participación ciudadana, si por ejemplo hay algún apoyo económico a las personas, o son voluntarios, o cómo es ese proceso. I don't know if in English, but it's about the challenges.
Challenges about citizen science. Muchos, a lot of. Empezamos con el objetivo de recopilar información y nos dimos cuenta que la gente estaba más preocupada porque le demos una respuesta que verdaderamente por el dato. Entonces, ahí fue cuando necesitamos articular con el Ministerio de Salud, y si bien sirve la información que recopilamos, la respuesta era lo más importante. No, la colaboración comunitaria, participativa o ciudadana es voluntaria. Tratamos de tener buenos canales de comunicación, hacer conocer a GeoBin, que sea una herramienta que sirva, y al mismo tiempo los agentes sanitarios que la empezaron a usar la llevan a la comunidad, y la comunidad las usa y articula con ellos en cada provincia de Argentina. Y eso es lo que vamos a empezar a hacer ahora también en Minas Gerais, en Brasil. Así que, pero no hay plata. No, todo voluntario.
I actually messed up my own talk because it was my turn to talk last time, and you probably already noticed. Soledad went before me. Okay, I'm here as just a representative of the GBIF task group on vector-borne diseases. I was there as the data scientist of the group, and there were many other people involved in this, and I'll show you who those were in a minute. But this group worked for 4 or 5 years trying to mobilize vector-borne disease data into GBIF. And you probably know already, but I thought you'd just repeat the vision of GBIF, which is a world in which the best possible biodiversity data underpins research, policy, and decisions. So I should be timing myself. And also, GBIF is about mobilizing those biodiversity data, and their mission is about mobilizing those data. And it was seen in GBIF, and the science committee, I guess, decided that it would be a good idea to make a particular effort on vector-borne diseases because of all those aspects of policy and human well-being that are related to vector-borne disease. So these were all the people that were involved in the task group. As you can see from the flags, it was very global in outlook. There were people, I should note here, for instance, we had Florence Fouquet from the World Health Organization involved. She was there throughout the whole of 2 different sessions of the task group. I should mention also Paloma Chimbaru, who's from Brazil, but also worked in GBIF, and she was there throughout the whole thing and was really a powerhouse behind us as well. And I should also mention Dmitry Shigel from GBIF, who kept us all together and in line as well. We had 2 rounds of calls for data papers on vector-borne diseases. This was an editorial on those, and those are all people who contributed vector-borne disease-related data. I think that was very successful, and I'll show you something about what we managed to publish. 670,000 occurrence records into GBIF of vector-borne disease-related data, 890,000 specimens. This was all published with a CC0 public domain waiver from data from more than 70 countries, and we had mosquito data, sandfly data, ticks, kissing bugs, but I can't pronounce that. Snails and rodents, obviously disease vectors, rather disease reservoirs rather than vectors, but they were also important. And you can see from our summary data of that, you can see we covered, for instance, mosquitoes from North and South America, from Europe and Southeast Asia, rodent populations in West Africa, sandflies from South America. We have the kissing bugs also from North and South America as well, and ticks from North America too. We also contracted Francisca Astorga from Argentina. I guess you may know her, I don't know. But she did an analysis of GBIF data using of vector-borne disease studies, and separated studies that had used GBIF-mediated data from those studies that had not used GBIF-mediated data, because she was trying to see what people were really using. And if you separate those studies by disease that they were researching on, you can see some clear differences. For instance, it's quite interesting that viruses are quite high on the GBIF-mediated data. So there's not much virus data in GBIF, but that's because a lot of the host data is in GBIF, and so that's why that's there. For malaria and leishmaniasis, it's a bit more even. Malaria because there's a lot of mosquito data in GBIF, leishmaniasis because there's a lot of sandfly data in GBIF. But it's also interesting to see that for Zika and yellow fever, it's almost all GBIF-mediated data, and it's based on the hosts. So if you look at the hosts, you can see, yeah, oh, I should explain. These two guys are back to front. So that one is GBIF mediated data. That one is GBIF mediated data. I didn't have a chance to switch it around. So yeah, all the bacteria, viruses, and parasites are coming from non-GBIF related sources, very little coming from GBIF mediated sources. But when you look at things like mammals, obviously we have a lot of data in GBIF on mammals, and that's why GBIF is strong on those. It's also very interesting to look at the source of the data, where things are coming from. So GBIF contributes a lot to the host and the reservoir population data, and also to the vectors, things like mosquitoes and sandflies. But scientific literature is the biggest source. And I don't know if many of you have been to the talks yesterday about extracting from literature. Also, Anna was talking about extraction from literature. There's a massive amount of data hidden away in scientific literature that needs extracting to be able to do these kind of studies. And as you can see there, that contributes not just to the data on the pathogens themselves, but the host reservoirs and the vectors as well. And that's kind of interesting. And of course, there's quite a lot of health data, particularly for the pathogens themselves that are coming from other data sources. So we also organize trainings associated with meetings, for instance, like the AMV meeting in Thailand, I think that's right, yeah. And for training for vector-borne diseases. We have one of the participants here as well. And there's just a few other people. I'll just point out Paloma there because she was one of the most important people as well. I'll mention her later as well, because she published and wrote this report in GBIF. You can find it on the GBIF website about giving guidance to publishing data on vector-borne diseases and their hosts and pathogens as well. And I will stop there, but if you have any questions, if you want to know where any of that data are and how to use it, then please come to me. I think Kate Ingeldorf is here as well, and she knows a lot about this as well, and was also involved in the task group I forgot to mention before. Okay, thank you. So, any questions? We can always do this afterwards as well.
To know if after having extracted all this data from the literature, have you been able to contribute it to for a global data source where it's easier to access?
So Francisco did the literature. She wasn't really extracting the data. She was doing a review of the papers and deciding whether the data was coming from GBIF or not and what data they used. So yeah, it would be really good to go back over her corpus and then actually find where these data are and try and mobilize them. Okay, if there are no more questions, then the next person up look is Timothy, who's online, is recorded. Oh, you're doing, yes, sorry, yeah. Francis is Timothy now, yes. Yeah, exactly.
Oh, I need that. So hi again, everybody. So like you have noticed, Timothy couldn't be here today. So I'm going to do my best to do his talk and represent him. So that's why also I have a scarf now, because Tim likes scarf. So if you imagine I have glasses, maybe you will think that I am Timothy. So this work is part of, again, the GeoBond One Health Working Group, and also the Verena Consortium. And this effort is motivated by the call of the quasi-apartheid organizations, like I said before, with the One Health Joint Plan of Action, that we can use ecological expertise for health studies or health concerns. So first, there's a recognition, again, that disease surveillance is a biodiversity problem. Since the COVID-19, there's a really big recognition that our health depends on the health of animals and the ecosystem. For example, more than 70% of emerging infectious diseases result from zoonotic spillover. So we need to know where species are, where potential hosts are, so we can better manage our health. Also, we need to know how the distribution is going to change with time. So we know that climate change is redistributing species and their pathogens in space. And biodiversity is threatened by human impacts, and changes in land use create more human wildlife impacts. So there's a big need to model where pathogens will be in the future to better adapt and be ready for future pandemics, for example. So in this presentation, I'm going to present 3 lessons that we learned from the biodiversity domain that can be applied for biosurveillance. And what I mean by biosurveillance is just the monitoring of animals and plants and their pathogens. So everything I'm going to say is part of this article. So I suggest to you to read it. It's only 2 pages, so it's quite short. So the first lesson is that variables, so EBVs, are really useful for monitoring biodiversity. So EBVs are essential biodiversity variables, and they bridge the gap between raw data and indicators for policy assessment. So in EBVs, there's different categories, for example, genetic composition, species population, species stretch, etc. And one of the tasks of the GBIF One Health Working Group is to try to identify which EBVs could be used to calculate indicators related to zoonotic diseases. The second lesson that we learned is that data clearinghouses are very useful. So Quentin talked about GBIF and how they can be used to analyze or store data about host and pathogens. And in GBIF, it's really good because they have widely accepted data standards, really strong community practices on data analysis. It's also part of the education curriculum, and also has multilateral and open governance. So the biosurveillance community can inspire or take that as a model to eventually have a better centralized data sources. The third lesson is that biodiversity monitoring happens across scale. So I don't know if you saw the other talks about the bonds, but biodiversity monitoring could be work with bonds, so biodiversity observation network, which is different collaborations between different stakeholders at different scales. And there's also, if we integrate all of these bonds, we can have a global biodiversity observation system, which can be very useful for monitoring biodiversity. But also, the biosurveillance domain can also get inspired of these core principles to eventually better survey pathogens. So what's next? So we can have studies that both study the locations of reservoirs and also host virus interactions. So here, Michael is the author of this study. Hi, Michael. So what he did, maybe if you have questions about it, you can answer later. But what he did is that he used SDMs to predict where reservoirs are. He also used host virus interactions to try to identify places where we should sample to optimize or minimize the uncertainty about hosts and their pathogens. So this is an avenue that integrates both fields. Also, a practical tool is Pharos. I don't know if you've heard of it, but it's a database of host virus interactions, specifically about mammals. It is based on fair sharing of wildlife infection data. It also includes negative data, so it means that animals that were tested negative for pathogens. That's a rich data specification, so a standardized vocabulary. It is multi-purpose, so it can be used for biodiversity conservation and human disease surveillance. And it's also compatible with Darwin Code and Humboldt Core. It's also based on the WDDS data standard. So I think the next presenter, Conan, will talk about this further. Finally, another tool that can be useful for biosurveillance is Banana Box. We have Jean-Michel here for Banana Box. And Banana Box creates pipelines created by the community to optimize or better calculate indicators of biodiversity. But these pipelines can also be used for biosurveillance, and this is something that we're currently working on. So take-home messages. There are shared practical challenges between biosurveillance and biodiversity monitoring. For example, there's a lot of things to monitor, but not enough resources to do so. However, there's unique challenges for biosurveillance. For example, there are more regulation on access to information, so the data is more close usually. So sharing is caring. We need to put resources, technology, and good practices in common and not reinvent the wheel. And we need to think about integration and synergies. that wildlife disease observation networks can be built on top of biodiversity observation system to really better integrate these two fields. So I don't know, Tim, if you're online, but I hope it was okay. And you can scan the QR code if you have questions, or if you, not if questions, if you want to see more about what we do in the lab. So thank you.
Thank you.
Yeah, any question?
Okay. Okay.
Yeah.
So I think the next talk is online, isn't it? Yeah, and so we'll go straight over to that from Colin Schwantz. To mute this for now. I hear them.
Identify potential reservoirs and look for gaps in data collection. But as Given et al. Demonstrated, biodiscovery rates are highly dependent on sampling and data availability. If we look at the graph on the right, we show the number of viruses discovered per year by million order. We can see that for the majority of orders, viral discovery is accelerating as sample effort increases. This indicates an incomplete knowledge of host-virus associations. Researchers are actively using biodiversity data to fill those gaps. Given et al. Use a machine learning model to identify museum specimens at the Field Museum of Natural History that could contain coronaviruses or paramyxoviruses. They tested thousands of samples from bats in the collection and found 3 novel associations for both coronaviruses and paramyxoviruses. When it came time to publish their findings, it was clear that sequences from the viruses detected in the bats should be deposited in GenBank. And the museum specimens were already in GBIF. But what should they do to link those data? How do they report negative results, testing protocols, or other critical elements of the study design like model predictions? The domains of micro and macro biodiversity data have separate, mature data repositories with robust and commonly used data standards. However, when a disease ecology researcher has to link those domains, unifying frameworks are difficult to find. The Wildlife Disease Data Standard unifies those mature systems. It was designed by a diverse set of members of the Marina Institute. The data property in Woods draw on the data standards from each domain to put equal emphasis on micro and macro biodiversity data, as well as reporting of the presence and absence of host pathogen interactions. This combination facilitates the creation of fair data sets with high utility for pathogens across the One Health spectrum. Woods draws on terms and identifiers from occurrence data standards like Darwin Core and nucleotide genetic sequence databases like GenBank. It leans heavily on DataCite for metadata related to citation and retrieval. These 3 pillars allow Woods to connect data that may be spread across GBIF, GenBank, or Zenodo, and increases the chance of discovery. Woods is a minimum data standard that can be extended. As long as users don't remove required fields or change field types, their data should be valid under the generic Woods schema, meaning it can still be aggregated with other Woods compliance data sets. If, for example, a researcher collected acoustic movement and serology data from bats, Woods could be extended to include links to the data in those respective repositories. The standard can also be refined. Researchers can do things like use a controlled vocabulary to describe its life stage or set up range limits for specific measurements. WIDS is intended to be accessible to a range of practitioners, from those working primarily in spreadsheets to those building complex data infrastructure. The standard itself is written as JSON schema, but the repository that it's stored in includes templates and example data in Excel or CSV formats. To make it even easier to use, we've developed an R package called WIDS Wizard that has convenience functions for aligning data to WIDS using different versions of the standard and validating data. The package also includes a set of vignettes explaining how to incorporate WIDS at various points in the research data lifecycle. I'm hoping to have a Python version very soon. Conclusion, WIDS fills the liminal space between established biodiversity data infrastructure and offers one health practitioner a viable path to sharing data sets that combine occurrence data and human data. WIDS allowed June et al. To report negative results, testing protocols, and model predictions via robust metadata that links the primary domains of biodiversity data. The WIDS Wizard package made that process relatively painless. We hope that WIDS serves as an example of a practitioner-focused data standard that encourages sharing and linking biodiversity data across scales. Thank you all for your time. This work was funded by the NSF, conducted at the Yale School of Public Health.
We have Colin online, or?
I am here.
Okay. Hi, Colin. Thank you for your talk. Do we have any questions for Colin? But I did have a question for Colin myself. So we've been hearing a lot this week about the Darwin Core new data schema, and I just wondered how that could be used with WIDS potentially to make it all more interoperable.
Yeah, that's a great question, and that is something that we are actively thinking about. Right now, we have vignettes that show users how to convert data into a frictionless data package. So taking that one step further and making it compatible with the Darwin Core data package should be fairly straightforward. It's on the list of GitHub issues right now.
Great. So Kate Ingeldorf just said it's on her list too. Okay, if there's no further questions, I think the next one is online too, or he's not here though. At least I haven't seen him. We haven't seen him, have we? No. I think he's not got a recording. Oh yes, there's a video. Yeah. So just while we're waiting, I could mention that I've been involved in a BAT group for 5 years, is it, since the pandemic? Donat also from Blasi, and Anna Willoughby as well, and also Nate. Here we go. So if you want to know anything about BAT data and you would like to be involved in that group, then please do come and see us.
Good morning to everyone at Living Data. Sorry, I couldn't be there at person. I'll be able to share these research results with you. This work is really quite a collaborative effort. We've got a lot of co-authors here. I'll talk some more about this international group that we've been collaborating with the last couple of years. And so really what we've been thinking about broadly, this idea of spillover risk, the data optimize that. So just to introduce that concept, you can map it onto phylogeny. In this case, thinking about spillover from other mammals to humans. We also think about spillback from wild mammal populations. And then you can visualize this in other ways. So, for example, the bipartite network is a common way to put this in. And really, one of the questions that we've been interested in is not just between the bats and viruses, but between the bats themselves, co-roosting interactions, other ecological interactions. For example, like if two bats are nesting in the same cage, how might that interfere or alter a given host virus? We might imagine that bats that share a cage might be more likely. Really, what this kind of gets at is this idea of taxon-specific zoonotic risk. This idea that some taxa, like bats, might actually be more likely to host viruses that are going to be dangerous to humans, zoonotic viruses. So this has been called the bat reservoir hypothesis. This idea that bats are special reservoirs of human viruses. But there's this other idea too that there's lots of bat species. There's a lot of host species richness, and the viral richness might just scale with that bat species richness. In which case, we'd actually expect rodents to have more viral richness because of more rodent species. That's a big moment. So really, this question, while interesting, is not easy to address, in large part because this kind of global scale viral is really not very resolved. Biases associated with it. That's kind of what we wanted to take a look at in the context of taxonomic effects, biodiversity data in general, and we were familiar with, but wanted to see how it's impacting this kind of viral risk. So you have a given species here that has a type specimen and a name associated with it, and then we learn more about the biology of these animals, and a taxonomic split occurs. That type specimen stays with the name as it would, and another part of the taxon gets split out here and gets a new name of B. So you end up with this sensu stricto versus sensu lato concept of the same species name. It's really a common thing that happens, but it messes up this kind of trait data or interaction data between, in this case, bats and viruses. And so, yeah, really these are kind of the questions. When do these taxonomic splits change viral risk estimates? Like, at what scale is this question? Does this really matter? So this is something that, in terms of tracking taxonomic change, that I've been involved with at the level of all mammals. Thinking about global mammals, there were the volumes of mammal species of the world. IUCN Red List was an important component that was closely tied to MSW3. And really then the important thing here was that these geographic range maps were generated for the first time on a global scale. Subsequent to that, other versions of IUCN have come out that have modified those maps. And then in 2018, we published the first version of the Mammal Diversity Database, and we've subsequently done other versions of the database that are tracking just kind of taxonomic change through time. An important thing that happened in 2022 was to, for the first time, publish range maps that were not just the IUCN range maps. So these other versions of range maps tied to the Handbook of the Mammals of the World, Illustrated Checklist, and the Mammal Diversity Database. So this version 1.2 of the Mammal Diversity Database has range maps that were published in that article. And then my now former PhD student, Angel Robles, and visiting scholar Norma Hernandez worked on updating these range maps to version 1.11 is kind of as far as we've gotten so far. And there's this MDD maps R package that you can use to access these. So then, yeah, basically we have these kind of different periods of time where we have the initial IUCN maps until the first version of the MDD maps, and then the 1.2 to the 1.11. And we can kind of look at the impact of taxonomic concept change through those different time periods. So how do those range map changes impact bat virus interactions? And really, we're talking about taxonomic splits and taxonomic lumps that we can track with the use of these range maps. And so the kind of cool thing here is looking at the amount of change that's happened, right? So in that first period of change, 185 species of bats were affected by either a split up to the new species or affecting an old species. So that's 12% of bat species globally. And then just during this 3-year period, another 60 species. So we have, 16% of global bats, or about 1 in 6 of global bat species was affected by some taxonomic split since 2008. Let that sink in. That's quite a bit. We have this kind of activity going on, and we want to have it going on, but we want to do a better job at tracking the impact of those splits on biodiversity data in general. And I think the viral interaction data is a high priority type of biodiversity data. So most of these splits were dividing 1 or 2 species from an existing, but some of these splits were dividing out 3, 4, 5 species. And so then the kind of really interesting thing is to then intersect for those species, how many viral interactions are known kind of from these different sources, and then what's the impact on those. So we use this Virion data set that many of you are probably familiar with. There was an effort in that Virion data set to harmonize the taxonomic names with NCBI taxonomy using synonyms. So there's that kind of like artifact to think about here. These are not necessarily the verbatim names from the data sets. But then from that, what we ended up finding is that those 245 taxonomic splits impact about 22% of all the bat virus interactions in the data set. And these are the top 15 species of bats that have ambiguous viral interactions, including up to 400 in Miniopterus schrebersi, which is one we'll take a look at here. And so just thinking about this geographically, this is a really widespread species, or was considered to be, and kind of grouped with Fulginosus in a taxonomic sense. And then with these splits, we now see that this is like completely kind of rearranged. Schrebersi is now only in Europe, and we can tease this apart. So the split, this is these are the top 20 viruses, kind of breaking down those 400, including some bat coronaviruses. And we look at this on GenBank. We should be able to tease this apart if that interaction had a really good geographic locality. And we see that, for example, this bat coronavirus has a geographic locality of China in 2012. That's really broad. And so if we look at China now, actually this bat species is not considered to occur in China anymore. So now we need to go back and dig a little further. Like it might not be able to be known if no voucher specimen was taken at the time of that bat virus interaction, right? It's just, that's what they said it was, and now we don't really know. So there's a broader issue for mammal virus data in general. This kind of taxonomic curation is needed. There's sort of a decay process that's happening of ecological interaction data in general. And then really what's needed to unlock this is looking at the context in the literature, right? So we need to be not only digitizing data, but connecting it and connecting it back to its source in the literature. This is, like, as I mentioned, a really broad collaborative effort with a lot of excellent scientists that have made this possible, some of which are in the room today. I know Donat and Quentin are there. So again, sorry I couldn't be there, but really would appreciate feedback, and thank you all for your time.
actions. What about the effect of sort of cryptic species? Is that an issue? Or cryptic species complexes?
Sorry, is that question directed to me?
Yes.
Okay, yeah, so you're thinking morphologically cryptic species. Right, yeah, so that's one of the issues if the bat virus interaction is just being identified morphologically in hand, that's obviously an issue. In some cases, there's DNA of the bat host that's also being taken, and then genetic identification can be made. But really the critical thing is that those bat virus interactions need to be associated with geographic metadata. That's really what kind of future proofs given interaction to taxonomic change of the type I'm talking about here, where you have a concept change. And that's, if that metadata was taken, it's often not kind of following the interaction forward through these databases. And so that's really what we need to be paying more careful attention to.
Okay, thank you, Nate. There's no more questions in the room as far as I can see, but I hope you'll hang around to the discussion because there might be still more things there. Sorry, next up we have Luz Dali. Hey.
I'm going to speak in Spanish. Mi nombre es Luz Dari Acevedo Sendales, de WCS Colombia. Pero esta presentación es de Diego Montesino, quien no pudo estar acá. Entonces voy a hacer mi mejor esfuerzo para explicar este ejercicio que viene haciendo WCS en diferentes partes donde tenemos programas actualmente.
Sorry.
Ah, ok.
This one.
Yeah. Bueno, pues el principal contexto como surge este ejercicio es a partir de los diferentes programas donde hemos tenido acceso a trabajo con comunidades y a problemáticas de conservación específicamente, que es como ha nacido el trabajo que hacemos de campo, como es el Field Program de WCS. Entonces, tenemos ejercicios en diferentes partes donde nos enfrentamos a situaciones que no son solamente de atención a brotes de origen infeccioso, sino también a temas, por ejemplo, a la salud de la vida silvestre, como la cacería, ¿sí? O como problemas que tenemos en las interfaces, sobre todo interacciones en los bordes, donde tenemos fauna doméstica y fauna silvestre interactuando. Este es un ejemplo de lo que sucede en Costa Rica actualmente, y por ejemplo aquí en Colombia tenemos el problema hace 5 años, donde el gusano barrenador se ha extendido y es una problemática que ya está cayendo sobre especies silvestres, como en este caso en Colombia, tapires, y ese es el caso de Costa Rica, donde también, además de tapires, está afectando otros mamíferos. Y casos donde, por ejemplo, tenemos muertes en áreas protegidas, muy esporádicos o a veces brotes donde tenemos muchos individuos involucrados. Ese es un caso en Camboya, donde enfermedades del ganado fueron transmitidas también a fauna silvestre. Este es un poco como los vacíos que nosotros identificamos para trabajar esta propuesta de base de datos, y uno es como que tenemos diferentes temas de salud en la vida silvestre y que son estos datos para poder revisar lo que está pasando, para poder priorizar, son tomados por diferentes actores, diferentes lugares, diferentes metodologías, y con esas diferencias es un desafío poder manejar los datos y sobre todo poder entenderlos, analizarlos y hacerlos de manera comprensible de tal forma que se extienda a otros actores que son los que toman las decisiones sobre estos territorios. Entonces, pues este contexto no es desconocido. Tenemos varios reportes y artículos que lo muestran. Por ejemplo, aquí quiero resaltar donde se hace una evaluación con diferentes profesionales y se pregunta cuáles son como los diferentes desafíos dentro de su ejercicio para poder atender esta problemática. Y uno de ellos es que pues en su jurisdicción o en su mismo sector es difícil compartir los datos. Por otro lado, también se propone si se pregunta si esos datos se pueden mejorar, y pues la respuesta de la mayoría de los profesionales es que sí se puede, pero todo está en la voluntad. Y por último, se pregunta si eso es posible mejorarlo en su territorio o en su área, y si es importante, y pues la mayoría siempre va a responder que sí, pues es como el dato que muestra en la última parte. Por otro lado, también estudios que muestran la dificultad del acceso, sobre todo a datos que son tomados por entidades del gobierno o por autoridades, y pues lo que hace difícil que estos datos sean compatibles entre diferentes plataformas. Entonces, como ustedes pueden ver, este es un resumen de lo que acabo de mostrar, y es, si tenemos necesidades de centralizar la información, tenemos necesidades de tener un lugar, un repositorio de esa información que sea accesible a la mayoría, y tenemos necesidades de guiar a partir de esa información para desarrollar estrategias y propuestas y priorizar. Entonces, la propuesta de la base de datos es un ejercicio, esta es como la propuesta, los ítems que hemos desarrollado con ejercicios en diferentes partes, y tiene una propuesta donde va como el objetivo, cuáles son las metodologías que vamos a utilizar, cuáles son los periodos, los lugares donde vamos a trabajar, el tiempo que se va a utilizar para el esfuerzo de muestreo, con cuáles taxones, cuáles son las muestras que vamos a tomar, y cuáles son los diagnósticos y los objetivos de ese diagnóstico para poder interpretar y que eso se transforme luego en ejercicios de gobierno. Entonces, este es un ejemplo, esto es como la base conceptual anterior, pero eso es un ejemplo ya, por ejemplo, de lo que tenemos aquí en Colombia, un ejercicio que trabajamos en un proyecto Bolivia-Colombia, donde estamos haciendo ejercicio con mamíferos, en este caso murciélagos y roedores, y cómo se organiza la información para que sea compatible entre dos países, con ejercicios con comunidades, y donde la información se está tomando de la misma manera para que sea compatible, interoperable, y que al interpretarse se pueda ver situaciones en diferentes territorios donde las condiciones son también distintas. Aquí estamos colocando, pues están también los vectores, pero al lado también estamos mostrando un ejercicio donde la información no está estructurada, y la base de datos permite que esa información, que no es tomada de manera tan rigurosa, por ejemplo por un guardaparque, también se pueda adaptar, aunque no tengamos toda la información, pero entonces se puede como arrastrar esa información y llevar para que sea compatible con el resto de información que suceda en un mismo territorio. Esto es de una manera programada, y allá en el ejercicio nos muestra algo que es en un patrullaje de un guardaparque. Entonces, esto es como se vería la base de datos, la estructura, es la misma información que acabamos de ver en el ejercicio anterior, en la anterior diapositiva, y al final lo que se busca, o cuál es el propósito de esta plataforma, es que tengamos una información compatible, que haya disponibilidad de datos, que podamos hacerlos interoperables, que haya protección de la información dependiendo de quién la esté usando, y que esos datos, que haya un repositorio en un solo lugar donde esa información se pueda centralizar y pueda ser compatible con diferentes organizaciones, entidades, en un territorio o a nivel global. Entonces, como conclusiones, está que la información pues, es accesible a cualquier entidad, se puede tomar por parte de cualquier entidad, o puede ser leída y tomada en cuenta por una entidad del gobierno, una entidad privada, una organización o la academia. Que la estructura de los datos pues, sea compatible, es también con plataformas a nivel global como el GBIF, o por ejemplo la que maneja la Organización Mundial de la Salud para la salud animal. La También que los datos estén centralizados y asequibles. Y por último, que esa información sea asequible a tomadores de decisión en un territorio, por ejemplo, como comunidades indígenas o campesinas. Para ellos, por ejemplo, la plataforma lo que busca es que este tipo de actores no paguen por el acceso a la información o por ponerla en este mismo repositorio. Y la idea es que también el usuario tenga el acceso a la información o que, por ejemplo, si quiere no compartirla, pero usar la plataforma, se puede también usar de esa manera. Y por último, tiene la posibilidad de que se pueda manejar en diferentes idiomas. Entonces, por último, esta información, este es el artículo que presenta la plataforma, se publicó mismo hace menos de un mes, y toda la información, todas las preguntas que tengan se las pueden hacer a Diego en esos enlaces. Igual, si puedo responderles con mucho gusto lo haré. Y bueno, esto es un ejercicio en el que han venido trabajando en varios años, en varias organizaciones, pero la idea es que sea compatible, que podamos nosotros orientar sobre cómo tomamos la información en la vida silvestre, porque es todo un desafío, pero que esto sea compatible sobre todo con otros sectores, como la Organización Mundial de la Salud o como la Organización Mundial de la Salud Animal, para que entre diferentes sectores podamos tomar decisiones a nivel global, regional o sobre un territorio muy específico. Muchas gracias.
Es muy impresionante. Do we have any questions for Luz?
Spanish is okay or okay, gracias Luz Dario, pues súper interesante la.
I'm Leonardo Biotti.
Oh, just. Ok, gracias. Muy interesante la presentación que hiciste, me parece que es súper importante porque, pues no sé si todos saben, pero en Colombia tuvimos este año un brote tremendo de fiebre amarilla y entonces teníamos muchas dificultades para acceder a información, por ejemplo, de la ubicación de los primates, ¿sí? De Aluata, bueno, los que están como relacionados también, porque sabemos que en la fiebre amarilla, cuando empiezan a morir los monos, inmediatamente es como una alerta epidemiológica para entrar en acción. Entonces, me parece muy cómo tienen orientado, no sé, hacer difusión de esta plataforma o cómo se puede acceder. Decías ahorita que pues hay unos usuarios que tienen algunos permisos, pero esta información, digamos, en un momento así de contingencia ante un brote es súper importante porque a veces trabajamos todos por separado, ¿no? Entonces está la salud por un lado, está por otro lado pues la parte de conservación, pero en esto pues tuvimos mucha dificultad con eso y me parece muy interesante. Gracias. Ese es un desafío. Nosotros precisamente cuando apoyamos trabajos con comunidades o con gobierno, es algo que siempre vemos y lo hemos visto por mucho tiempo y por ejemplo aquí en nuestra realidad, lo que tú dices, Latinoamérica está llena de ese tipo de situaciones. Ahora, creo yo que, por ejemplo, ese ejercicio, o sea, ¿cómo va a ser el acceso? Estamos en este momento haciendo pruebas con diferentes proyectos. Les mostraba el de Colombia, Bolivia, porque es algo que precisamente buscamos que la base, la plataforma, se adaptara al proyecto, no el proyecto a la plataforma, para que precisamente empezáramos a ver cómo crearla y cómo tiene que ser sencilla, o sea, con muy pocos datos y también empezar a ver qué datos estamos dejando de tomar para que después los tengamos en cuenta a futuro para prevención o para control. La manera en que va a ser asequible puede ser a través de la organización, o sea, nosotros y también los otros actores que están involucrados en los diferentes países, pero en este momento el liderazgo sí lo tiene WCS. Yo te puedo poner en contacto con Diego directamente, él es chileno, entonces puedes hablar con él directamente. Y también hay algunas herramientas que tienen que ustedes pueden ver cómo funciona en los dos enlaces que les mostraba de cómo funciona para que entiendan. Hay un manual donde explica cómo se está pensando organizar y lo que hay que ver es cómo su proyecto o su situación se tomaría la información para que no quede como suelto y la forma en que se analizaría, que es una gran ventaja, es que ya está todo, simplemente tú metes la información y te ayuda a analizar
o te ayuda a orientar qué tipo de información puede estar haciendo falta de otros sectores para hacerla interoperable. Gracias.
Yeah. The complexity is enormous, and yeah, the amount of organizations and people that have to be involved and the transsectorial nature of the problem is very big. So next we have Leonardo Viotti, and he's got a recorded. He's also online. Great.
Hi, thank you for coming. I'm Leonardo Viotti, and believe me, I'm not happy to be doing this virtually and not be there with you in Bogota. But even if virtually, I will present some of our work in mapping priority areas for biodiversity conservation with the goal of preventing zoonotic spillover. Well, this work is done in collaboration with all of these very smart people. And while the credit is certainly shared, if there are any mistakes or inaccuracies, the blame is very likely to be mine. Here's a quick summary of what I'll cover today. First, I'll do a quick review of the literature and tell you why biodiversity conservation could be a good strategy to prevent disease. Then I'll show the method we developed to map priority areas in high spatial resolution. And finally, I'm going to apply this method in tropical areas across the globe to show good candidate areas for conservation interventions and give you some estimate of how much this could cost. A little bit of context, this work is based on the idea of One Health, which is this concept that human, animal, and ecosystem health are deeply interconnected. And while part of this is due to other ecosystem services, a good chunk of it is because it turns out humans are also animals. So other animals can transmit disease to us, and there are certain conditions that can make this transmission much more likely. To start, diseases originating from wildlife cause substantial human suffering and economic loss every year, and it is estimated that 2/3 of all diseases affecting humans have origins in other animals. And examples are the coronaviruses, HIV, influenza, but there are many other examples. And there's good scientific evidence that healthy ecosystems can reduce the likelihood of disease transmission. However, it's not immediately obvious how to do so and where. And in part, as a consequence of that, disease prevention has largely been neglected in conservation efforts, and conservation has been largely ignored in disease prevention efforts. It has been shown in this 2022 World Bank report that prevention can be much cheaper than mitigation. So what is a zoonotic spillover? Zoonotic spillover is the event when a pathogen jumps from an animal reservoir to a human host, and it often involves complex interactions between humans, their livestock, and wildlife, and it may or may not involve the presence of a vector such as mosquitoes and ticks. And why do we believe biodiversity conservation could be an effective strategy? Well, the literature shows that in healthy ecosystems, there are a lot of barriers preventing these events to happen. Many factors have to be aligned for a zoonotic spillover event to take place. However, deforestation or other land use change that causes environmental degradation starts to release these constraints and increase the frequency of which these events can occur. First, it promotes the overlapping distribution of humans and disease reservoirs. Also, the stress factors can compromise the fitness of animal hosts, which can promote sharing of pathogens, which is how many pathogens are these animals releasing to the environment. Also, changes in the environment can lead to changes in composition of species, and there's good evidence that species that are more resilient to environmental change also tend to be more resilient to disease. So if there's a patch of forest with, let's say, 10 species of bats, and it's cut down, the 2 or 3 species that remain, and now probably we push to rural areas or in more close proximity to humans, also happen to be more likely to carry very nasty viruses. Lastly, for an event to happen, we need direct contact, and certain human activities can promote that either through hunting, collecting guano, for example, or raising livestock, particularly in high densities in areas close to wildlife. So what we're advocating in this paper is for ecological countermeasures in two different fronts. First, preventing deforestation to happen in the first place, but also restoring already degraded areas. So the key idea here is that less environmental degradation, less disturbance, the less likely spillover events are to occur, which great, but where and how can I do this? We developed this methodology to flag areas where well-documented risk factors align, and we do this with publicly available data, and we provide these layers at a resolution of 1 square kilometers for tropical areas across the globe. The layers we are overlapping is high biodiversity of key taxa. These are bats, rodents, primates, and birds. Also, high livestock density, especially of pigs and chickens, high human population density, and two proxies for environmental degradation. One is deforestation risk in Q 2050, and the other is restoration potential. Also, we combine these with two additional layers for cost-benefit purposes. One is the existence of protected areas, and also the opportunity cost of the land. So we combine these into two layers of overlapping risk factors. First, areas with high density of livestock and high density of humans, and also areas with high biodiversity under the threat of deforestation or already degraded, but with the potential to be restored. We further combine these into two types of priority areas based on the type of intervention that we think should happen: areas with high livestock risk and high biodiversity, and where under high risk of deforestation, and areas where ecological restoration could be made. When we apply these to tropical areas across the globe, we find around 200,000 square kilometers of areas where all of these risk factors align under the threat of deforestation, of which approximately 95% are outside of existing protected areas, as well as 275,000 square kilometers of areas with the potential to be restored. And if we were to implement this tomorrow, the opportunity cost of the land would be around $2 billion in perpetuity, which is a lower bound. However, to give you a sense of scale, a previous study has suggested that just reducing the likelihood of extreme disease outbreaks by 10% could reduce deaths by 300,000 and save $2 trillion each year. Here is the distribution of priority areas under the threat of deforestation when we apply our methodology to tropical areas across the globe. And the idea here is to flag areas of potential intervention where deforestation prevention could be an effective strategy when trying to reduce the risk of zoonotic spillover. Similarly, here are areas, priority areas with high restoration potential. And one thing to flag is that our idea is not necessarily to be prescriptive and say you should do these interventions here, but rather to propose a methodology that could be adapted to local contexts. And in order to do that, we provide two sets of tools. First, we provide this no-code application on Google Earth Engine apps, where you can replicate our analysis and fine-tune the parameters to your own needs, and you can access it through this bit.ly link below. Second, not only all of the data is publicly available, but also our goal is to make this completely open source, so you can download our code and redo this analysis however you choose, changing the layers, the resolution, everything. And this code should be published soon alongside a preprint. So here's some of the takeaways. So ecological countermeasures can be a very cost-effective strategy for reducing the emergence of disease. However, our study shows the extent of which conservation efforts currently fall short of this goal. Thank you so much. Please don't hesitate to reach out if you have any questions or any comments, and you can find me on my email below. Thank you so much.
Thank you, Leonardo. We already have a question, so I'll just from Alice Hughes.
Thanks for the talk. This is clearly an important topic, but I'd like to know the justification behind the choice of mammal layers. The Jenkins and PIM data is not only over 10 years old, but it's based on IUCN data and a very coarse resolution. Also, different vectors are going to have very different ecological niches. So putting them all together may smudge out where relative risks are. So some rationale behind that choice rather than modeling species occurrences based on more recent data that better represents the intersection between where species actually are and the environmental factors behind that. Thanks.
Can you guys hear me well?
Yes, good.
Great. Yeah, thank you. Thank you for the question. This is a really great point. Yeah, so I believe we got the IUCN data we're using is a little bit newer than 10 years old. It is from 2018, if I'm not mistaken. And the reason for choosing that is a convenience due to the fact that's the only data source we could find that was available on such a scale. I would be if you have suggestions for like other data sets that could be globally representative or at least to tropical areas across the globe, I'd be very happy to hear that. But I think it's a limitation, especially given the lower spatial resolution. Yeah, we'll be happy to change this to other layers. This will be very trivial to change in our current analysis.
Okay, thank you. So, any other questions for Leonardo? So we come to the end of all our talks now. We have some time for discussion. I think something like 20 minutes if people want to discuss things. I have a few things I've noted down that we might be interested to discuss. If you have any topics you would also like to talk to, looking your direction too, maybe we'll start with that. That was actually last on my list, but I really don't have an order. Maybe you could introduce yourself and explain your project.
Okay, hi, my name is Jonathan Reddy. I actually work with DNA metabarcoding, and we've been working recently with spider webs as a monitoring tool. So we did a study where we compared spider webs to CDC traps, malaise traps, and pitfall traps, and they're extremely effective at getting the insect vectors and the potential host reservoirs. So I'm trying to put together a massive project, and I'm interested in partners, particularly from the Global South, who would be interested in taking part in that, to try and apply for some seriously big funding to try and get huge amounts of good co-collected data for zoonotic risk in general. So if anyone wants to talk to me, I'll be presenting a little bit of the results tomorrow in the DSI section, but I'm very happy to talk to everyone at any time.
Good, thank you. Okay, we have some questions online. So the first question is, are you considering getting your database indexed by GloBI? I guess that was to who? Oh. Also, Darwin Court A supports many ways to publish literature-based and parasite host data. You'll be better able to find many examples such as Darwin Court A, etc. That was for Anna, sorry.
So the question was about hi Yorit. The question about getting data sets into Darwin Core and GloBI. For our case, yeah, our data set will publish on Figshare, and so it should be readily implementable to put into GloBI. And then Darwin Core, right now our literature is on Zenodo, which has some integrations with species and other, I guess, geolocation taxa specifications for Darwin Core. So we use that integration.
Progress. Was there another question? Oh, for me. Question for Quentin, also from New York, Poland. It is well established that Darwin Way supports documenting host parasite associations, as many natural history collections and other projects do so. Are you familiar with this, and which of the data sets you've contributed use this ability? Also, as you know, GloBI is one of the largest index species interaction data, including parasites and pathogens. Are you familiar with the project? Why did you choose not to collaborate with GloBI? I don't think we didn't. So I know Yorit very well, and we've put data sets in with that. I think you're probably referring to the review by Francesca Astoria. We didn't actually extract any interactions in that. There were extractions in the papers, but it was a meta-analysis of publications. It wasn't really an exercise to extract. But maybe, Donat, I don't know if you were in the room when I gave my talk and I showed how much of the data that Francesca looked at came from publications. So a large amount came from GBIF, but a large amount of the pathogen data was coming from publications and been extracted manually. Maybe you want to say something about that? The Donat works for the Plasi organization, involved in extracting mostly taxonomic data, but there's apparently a lot of host parasite data in there too.
Yeah, we are specialized to take data out of publications, but we are not really specialized in this step to extract biotic interactions. But we have a collaboration with the Biodiversity PMC, which is a clone of PubMed Central, which includes something like 500,000 biodiversity publications. So it spans across the life science. It includes biodiversity publications and about 900,000 taxonomic treatments. And they are specialized in extracting biotic interactions. Yeah. And if you're interested to make you get into the system, you can contact us, and we'll help you to set up a repository for your publications you use for your research, which then can flow into the Biodiversity PMC. So you make actually a big service to the community, not only by providing access to the data you used in your research, but make it also widely accessible to a much wider community.
Hi, I'm Angelica Batista from Universidad Federal do Pará. A question for organizers, Quentin, Timothy, Colin. In the group you are managing to, are considering in the One Health the approach of the contaminants, pollutants, because they consider for food, fishes, and communities. One of the problems are the presence of mercuries and different contaminants, and also have an interest in the One Health. How it's managed in your group, or what is the approach that you are considering to addressing this open data and health challenges?
And that's a very interesting question, and one I can't really answer that well myself. Well, I would like to, but it's not. So I'm a botanist, but I'm very interested in and I'm very interested in the interactions between species and how that contributes to human well-being and things like this. But I don't know much about your subject. Anna, do you have any or Francis? Oh, I should microphone.
Francis, you found pesticides as one of the main areas that biodiversity indicators could contribute to. Maybe you have something to say?
Yeah, so it's not us who found it, but the pollution is clearly like part of One Health. It's also, there's a lot of actions about it to protect, to reduce pollution, to protect health. In our lab, we don't work on pollution, but we are still thinking about indicators that could be used to for these actions. Yeah.
how could we contribute from the groups that are already working with that? Because maybe we could do some collaborations in that sense.
Yes, I certainly think so. And I was thinking at the end of this meeting, maybe to just have a sort of sign-up sheet of people's email addresses. I can just connect people with each other, and if people want to, we can get it started. And I don't know who in the room might be interested, but we can see if that works. Good. There was a question over here as well.
I know this is a data conference, but I feel there's a very, very strong public relations component to this. And although the solutions that were highlighted were more preservation and keeping a distance via biodiversity, I wonder if it was addressed the way to convey this to the public, because the biodiversity equal risk association is a slippery slope.
Yeah, and I come.
It's not directed to anyone in particular, but
so I come more from the direction of the invasion biology community, and there's a very strong link between the activities of human beings and the introduction of new species and the spread of those new species. And I think that's why I have a sort of common feeling with One Health as well. And there's a lot of communication required on that, and understanding human sociology, and to understand why people are spreading things around. Many of the plants, for instance, and as a botanist, many of the plants people have introduced were done so because they want to use those plants. They weren't just introduced by accident. And so you have to have a lot of communication and understand people's needs as well. And I think that comes into health as well. We saw a lot about the risk and the issues of livestock. And of course, that's people's livelihoods as well. And so it's very important to have good communications on why we're doing, or why we want landscape change, why we want to preserve forest for human well-being as well. That really answered your question, but you have to think of it. You can't separate the biodiversity from the people. That's my point.
Quentin, I could speak to that a little bit. I don't know.
Yeah, go ahead. We can hear you.
Yeah, I think in the context of the bat virus interactions, that's something that we're definitely aware of, because there's been a lot of negative publicity towards bats in response to COVID and whatnot. And it's not unheard of that people would try and like exterminate bats like in a whole cave or something like that. But that would actually probably promote more interactions with humans that could be dangerous. So I think it's indeed a complex situation, because we want to be illuminating the data while also managing the risk and promoting positive stories about bat ecosystem services, right? There's obviously all the services of eating mosquitoes and preventing vector-borne diseases in that context. So yeah, I don't know, other than really trying to present both sides of the coin and trusting human populations to be able to deal with that kind of uncertainty. I don't know. I'd be open to other suggestions though.
So I wanted to just build on that discussion. For those of you who don't know or are online, I'm Alice Hughes. I'm a professor at the University of Melbourne, and I previously worked on bats in China and bats with COVID, which brings a whole lot of challenges all of its own. What we know is that spillover is most likely in highly degraded environments. No matter what set of vectors or intermediate hosts you look at more broadly, typically we see that the most competent hosts are in degraded environments, and that deforestation, as well as the loss of key elements of the habitat, are going to accelerate spillover risk. However, key areas where we are missing information is integrating species demography, ecophysiology, and change over time. One of the only systems for which there is good data at the moment is the flying foxes in parts of Australia, where they saw that the intersection of changing climate and loss of habitat basically caused interactions between bats and horses, and that led to the spillover of Hendra. But we still have major gaps in the ecology of these species. What we do know is the best way to accelerate spillover is by both disturbing animals, so great case study for how to exacerbate disease risk. Look at TB in badgers, where studies showed that culls would drive an increase in spillover risk by accelerating the rate of individual movement and stress, and increasing individual stress and interface is where we are going to see loss. So clearly culling and the destruction of habitats is the best way to exacerbate risks. Conversely, if we are going to maximize the benefits coming from species, including ecosystem services around pollination of many crop types in the case of bats, and the control of insects, these are dialogues people can get behind. One of the key elements of a One Health approach is maximizing these benefits, as well as highlighting the importance of intact ecosystems and respecting the ecology of the species within them. That is something that we as a community can get behind, because we need that data to be able to manage these systems across space and time. But we also need to highlight that there are many positive consequences from valuing these species, but we will actually amplify disservices by continued destruction of habitats. So just a few thoughts from me, hopefully that will provoke some more ideas in this space.
Wow, that was a lot. Would anyone like to respond? Oh, sorry.
Hey, everyone. Regarding the biophilia conversation, there was a recent publication in Frontiers of Conservation Science specifically about promotion of biophilia and prevention of zoonoses, and there's some good case examples of how that's approached from the community level. And I think that that's something that, you know, as practitioners that we're just dealing with low resources to deal with community interactions. So I just wanted to pitch that forth as resources about like frameworks to think about community interactions with low resources, and how we're actually communicating risk within, whether it's grant-based projects or emerging kind of programmatic efforts, because I see that same thing too, working with zoonoses. It's just difficult to be able to communicate risk. So I just wanted to share that resource.
Absolutely. Yeah, Anna, you have a question or comment?
I think also a future frontier is, and that I saw with a lot of the gastrointestinal parasites, and then it's been common in the case of like COVID and Ebola, is that serosurveys show healthy people are exposed to these diseases in these locations and do not, succumb to disease. And so understanding the how people are living and like what is in our immune system that is able to either deal with low-level doses or, while these are areas of risk, there is also an immense amount of people living that are exposed and then do not have disease outcomes. And bringing in the human side, sometimes the biodiversity, we don't focus on that side.
Oh, one more line. So this is from Angie?
No,
oh,
this
no,
is
two.
from Joey.
All right, from Yorit again. Thanks for that. So this. Oh yeah, Yorit, do you want to speak that out loud, because that's a lot to read?
Yeah, thanks. Thanks for your presentations. Really cool to see that there's such activity in the host pathogen interaction realm. And one of the things I wanted to clarify in the question to Anna is that I was talking about Darwin Core archives, not Darwin Core terms in Zenodo. So it's really cool that you're using Darwin Core terms in Zenodo. There's a way to annotate Zenodo deposits with Darwin Core terms. But I was referring to the existing capability of Darwin Core archives to both include literature observations as well as documenting host pathogen relationships. And many projects already do this. So that was one thing I wanted to clarify. And the other thing for Quinten is that the question I was asking about the collaboration is really that I noticed there's, like over the last 10, 15 years, I've worked with many projects that have already manually transcribed data from the literature. And these are all being, or a lot of them are being indexed through GloBI, so you can more easily find them. And yeah, this is why I was a bit surprised why you decided to redo the work and go back to the primary literature, because many of these data sets are readily available and open for reuse. Yeah, so I'm curious to hear your thoughts on that.
Yeah, I'd have to look, but I'm not. Yeah, Kate Dingle's going to respond because she was also part of that. Yeah.
So I think the key with that is Francesca was specifically going through and I don't know what her methods were for finding the data that was not, for papers that were not using GBIF data, but for papers using GBIF data that was specifically going through our DOI and our literature tracking systems. But as far as the way that she sought the other data.
Francesca may have used GloBI to find those papers. I'm not really sure. So, Donna, do you want to say? We need to wrap it up now. Yeah. So.
Only briefly. So in the biodiversity literature repository in Zenodo, it's not just a dump of publications. The really value is that you can add metadata. And one part of metadata is, for example, to add all these biotic interactions using the respective vocabularies. So you can actually create exports, like JSON exports, and use that. And in this article, somebody said there is an interaction between X and Y.
Yeah, actually, Donut, that used to be the case, but after the upgrade of Zenodo, these are no longer supported. So I can share an issue with you because it would be great to reintroduce that.
Thanks. I have just one more question on a different subject.
So thank you, everyone. My name is Maria Zambrano, and I work here in Colombia in a small research institute. And I'm fascinated with this meeting. I've never been here before. And the reason is because I'm a microbiologist. And I know viruses and parasites would be categorized as microbes. But actually, what I find fascinating about these talks is I don't see any mention of any other microbes, which are so relevant in all aspects of One Health. And so I'm just wondering if any of you have considered integrating that aspect of the biological world, which is like the most diverse that we have, into some of your studies.
I think that sounds like a big challenge to all of us. And I'm looking around the room to see if there's anyone. I think that's a good place to finish on. I think that's an excellent question. I think that is a gap. That is a gap. There is another community out there that perhaps aren't represented here. I just wanted to mention a few more things before we finish. Maybe we can find a piece of paper to write email addresses on so that I can connect people at the end. I wanted to thank Catalina, who's been a real great help, reminding me of all the things I need to do and doing that, but also my other co-authors, Nathan online. It was nice to have Leonardo here. Yorit, thank you for coming as well. And is there anyone else I need to thank? There probably is, and I'm probably forgetting them. Sorry, Francis and Timothy as well. So thank you all for coming. I wanted to mention tomorrow morning is the invasive species symposium, and that has a lot of commonalities with this meeting. And I did submit an unconference on cultivated and captive organisms because you saw many times we don't have enough data on captive and cultivated organisms, and it's not shared very well, and it's highly relevant to One Health as well. So please come and support that unconference session. Okay, thank you.